All right, Mark, I do want to jump to you on this question, but I'd reframe it a little differently. So, one of the comments that we'd made or that you'd made during our pre-call for this panel was, we often talk about low double-stranded RNA quantities, right? But at the same time, I think there's still a nascence to our analytics, right? They may not necessarily be the most sufficient today for telling us exactly what low means, right? So
Yeah.
can you share some of the limitations that you're seeing in the world of analytics for measuring some of these common impurities?
Yeah, I guess maybe just to step back a second first is I think double-stranded RNA as an impurity probably has a bigger impact when we're thinking about RNA as therapeutics than it does as a vaccine. Um, we're looking at higher doses of RNA and trying to really boost protein expression with it as a therapeutic model. Um, and so I think double-stranded RNA as an impurity is a little difficult from an analytical standpoint, just based on what it is. It's not a single distinct molecule.
Mm,
It's a mixture of different molecular weight species. So
mm,
it's not something that you can simply throw on an HPLC or capillary electrophoresis and be able to get a nice sharp peak and get good
yeah.
sensitivity and specificity. Um, so I think this one of the standard ways, one of the current gold standards for measuring it's with an ELISA test,
Mm,
um, and um, it's pretty sensitive. I mean, in our hands, we're measuring double-stranded RNA or the absence of double-stranded RNA to thousandths of percentage. Um, but is that, I guess the question is that sufficient sensitivity, taking into account what type of molecule that we're looking and measuring, and in the vast diversity
mm,
of molecular weight sizes that the double stranded could come in. Um,
mm,
and I think the way we report it might, the easiest and most straightforward way to report it from an analytical standpoint is on a weight percentage.
mm,
But is that the right way to
mm,
actually be looking at this? Is it more appropriate to look at as on a mole percentage,
mm,
um, which is not an easy thing to do analytically? Um, so I think this is an area, at least in the therapeutic space, that probably needs a little bit more research and a little closer scrutiny.
mm. Stu, you're over there making some deep
Yeah.
thinking faces.
I well, I
I love
totally
them,
agree
by
with
the
Mark.
way. I
Yeah.
love them.
Yeah. He's making great points. dsRNA is something, we've thought about for a long time and struggled with, the characterization of it, exactly what Mark
Mhm.
is saying.
Yeah.
It's not it's not a homogeneous population of molecules. There's different sizes and
Mm,
sequences. So again, one big limitation is sensitivity, like Mark's saying, and is the ELISA appropriate? Do we need to
mhm.
measure moles versus just mass?
Yeah.
The other question with kind of the way we measure dsRNA with the ELISA is what's the correct standard material or reference material?
Mm, yeah.
Because like I said, different sizes, different sequences. If you're using modified nucleotides, the antibodies will have different binding affinities to that dsRNA versus unmodified. So that's still kind of there's a lot of room for improvement, I think, on the dsRNA front. And we do need to be able to detect, very, very tiny amounts of it and characterize it better
Yeah.
because it is the main driver of toxicity in
Yeah.
the mRNA. So
Okay.
it's important. Yeah.
It still is important. So that answers the question. Is it really important?
Yeah.